Hi, everybody. Thanks for coming today. I hope you'll bear with me between Austin allergies. I used to live in Texas, and I forgot how bad my allergies are here. And a talk I did on Tuesday night, my voice is a little gravelly, so I sound like a man. Anyway, I'm Martha Carlin. I'm the founder of a company called the Bio Collective. I am what you call a citizen scientist. So 20 years ago, my husband was diagnosed with Parkinson's disease, and I had a background in business systems, and was a business turnaround expert. And I started to apply my approach to turning a business around to looking at Parkinson's disease. And so in my initial training, the way we look at a business for business risk, we actually map out everything that's flowing through the business, and we look at where the break points are. And so I started to do that with Parkinson's. And of course, what are the major things that flow through the system? Well, it's food and water and what we're putting in this tube. So over the years, I moved from nutrition, food, how we're growing our food, and starting to see the interconnectedness of all of this. And then in 2014, I read a book called Missing Microbes by Dr. Martin Blaser that talked about growing up in the age of antibiotic use and the destruction of something called the human microbiome, and how that was contributing to all these, what they called non-communicable diseases. And diabetes was one of those, but Parkinson's and Alzheimer's were in there as well. And so I was like, eureka, that's kind of it. Like, the gut is sort of your general ledger, if you will, from a business perspective. So I founded a company to start looking at the human microbiome, and I'm going to tell you a little bit more about what that is. We've already seen some great speakers talk about gut bacteria. But I was known as the poop queen. So my company is about 8 years old. For the first 4 years of my company, we had a little box that we sent to your house, and you sent us back your poop, all of it. And we turned it into what I affectionately call as, well, chitlets, only not chitlets, if you know what I mean. And we allowed researchers all over the globe to access those and do research on them, and then we did genetic sequencing ourselves. And along the way, then we built a system, a piece of software where we could understand how the microbes interact with each other and what they use and produce. And my ultimate goal was to basically restore our internal pharmacy. So, here, let me see if I can get this. So, what is the gut microbiome? Well, it's the trillions of bacteria, fungi, and viruses that live in and on our body. They're everywhere. And we are more microbial than we are human. Most people don't realize that. And since 2005, when they started studying the human microbiome, nobody had ever heard of it, really. You would have people who were microbiologists who were focused on bacteria, but most people never heard of the microbiome. When they started to do all this research on the microbiome, they did something called the Human Microbiome Project, and they have subsequently found that 90% of disease is connected to the microbiome. There are somewhere in the neighborhood of 2 to 3 times more cells from bacteria, fungi, and viruses in our body than there are human cells. There are 150 to 300 times more genes in our body that the bacteria, fungi, and viruses have than we have in the human genome. So they are functioning in a lot of ways as a primary system for us. 70% of our immune system is in our gut. So that's where this communication is going on at that thin layer of epithelial cells in our gut lining that is telling us what's coming in and how our immune system needs to react. So what do microbes do? I mean, in the last couple of years, mostly we've been made afraid of every microbe. There are a lot of microbes that do fantastic things for us. There are some that cause disease, but most of the microbes are, it's what they're doing, and they make everything. So even if you go look in the pharmaceuticals industry, the nutraceuticals industry, most of the products are actually fermented in giant vats of bacteria and fungi. They make enzymes, vitamins, neurotransmitters, short-chain fatty acids, peptides, antibiotics, flavors and scents, pharmaceuticals, and of course we know they make alcohol. So, what are some of the functions? I mean, I listened to Danielle's talk. She talked a bit about this. Those vitamin production is one of the main functions that goes on in the gut. The process of digesting your food, the microbes are producing enzymes and helping break down that food, and they're making the B vitamins especially, and vitamin K. So those are pretty critical. Fiber, breaking down the fiber. So I know on the keto side of the world, we don't eat as much fiber. But we do eat some fiber, and they're breaking down the fiber. And there are certain microbes that you need to have to break down fiber. And if you don't have those, you end up having some major digestive problems with fiber. You know who Hippocrates is? He said all disease begins in the gut, and everyone has a doctor inside. And really, that gut, our gut, is our internal doctor, and it's taking in that information from what we're putting down the pipe and trying to adjust to get us back to a healthy state. So these are just some of those diseases I talked about that have been shown to be associated with the microbiome. And I talk a lot about my husband, but my mother went to Mexico in her 20s and got the turista, is what they used to call it. But anyway, she got GI distress, and she had, back then they called it a spastic colon. Now they would call it IBS or Crohn's. So she had that, and it started this cascade for her where she went from all these digestive issues to having lymphoma, to then having MS, to then having breast cancer, to then having thyroid cancer. And all of that is this inflammatory process that is going on in the gut and can be traced back to certain microorganisms that produce toxins. So another key area that's driving the underlying problems in our gut is constipation or loss of gut motility. And they're they're related, but they're two different things. So constipation is typically more related to a lack of water homeostasis. So the opposite side of that is diarrhea, where you have too much water. Constipation is not enough water in the GI tract. And that's also signaling to these gut bacteria that are signaling to our cells that mess with the contraction of the muscles and stop the gut motility. And some of that is also related to not being able, if we're not properly digesting our food, then our gut will signal and hold on to what's in there to try to extract more nutrients. But what happens then is you end up with waste sitting in your gut. And that waste, a lot of those bacteria produce toxins. So then you have this toxic load you're trying to get rid of. Now, this is a highly technical slide, but there was a researcher who basically proposed this inflammation and post-infection sort of low-grade chronic infection in our gut that is affecting our tissues. And down at the bottom there with the little yellow circle is something called LPS. So LPS is the abbreviation for lipopolysaccharide. Lipopolysaccharide is the cell wall of a gram-negative bacteria. And those are some of the more pathogenic bacteria. So when you, through this process that your immune system is doing, it's trying to break down those and kill those bacteria, but the cell wall components are sort of floating around in your bloodstream. And that is an inflammatory marker that they actually use in animal models to induce diabetes, Parkinson's. Like you can go into the scientific literature and say, you know, is there an LPS model for blah, blah, blah? And it's incredible to me that they study this, but they're not asking themselves, well, okay, how's that happening in a human and how can we just stop that from happening? So this is just a few of those microbes and the kind of toxins that they're producing. I think a lot of these names you won't be familiar with, but most people have heard of botulism poisoning. So botulism is a neurotoxin. And, you know, these bacteria produce neurotoxins, so you can have neurological effects. So what happens over time is your immune system is getting sort of fatigued because it's having to deal with all of this, and you get higher and higher level of inflammation and oxidative stress. And that oxidative stress just keeps making things worse. You end up with this cascade that drives up over time to all these different diseases. So, you know, maybe it starts out with just, you know, if you've got a little bit of stress, you start getting some coagulation and aggregation of proteins. It starts to move along the cascade of these inflammatory markers, and you get heart disease, cardiovascular dysfunction, neurodegeneration, and diabetes or blood sugar issues underlie many, many, many of these issues because sugar feeds the bad bacteria. So this is why you came here, probably not for all that other stuff. You want to know, okay, like how are these bugs affecting insulin resistance? This lipopolysaccharide that I was talking about, this is what causes it results in something called metabolic endotoxemia. So you get this leaky gut you hear people talking about. This LPS crosses over so you can see the little gaps, and it's crossing over that very thin lining in the gut, and then it's getting into your bloodstream. And what happens when it gets into your bloodstream is it's raising all these inflammatory markers, and your body is trying to address that partly through insulin. In obesity, what you get is a shift in these different types of bacteria. So the Firmicutes, those are ones that make some of those toxins. So you get an increase in those toxins. You're getting a lower level of some of these Bacteroides that are beneficial, that kind of keep things in check. And then the actinobacteria, you get an increase in those, and those are basically bacteria that can produce antibiotics in your gut to kill other bacteria. So they sort of start taking over, and that shifts your enzymes and your ability to handle what's going on. So the altered microbiome then has that increased intestinal permeability, the increased lipopolysaccharide, and so you're getting all that inflammatory cascade, and insulin signaling gets impaired. So one thing that I don't have on the slides here, you see there's a number of people here talking about minerals and the importance of minerals, and digestion is a part of extracting the minerals from your food. Well, our food supply has gotten very, very depleted of minerals. The soil is depleted of minerals. That means the animals, the vegetables, the fruits are all depleted of minerals. And you you've got to have those minerals to make lots of things work in the body. But there's one mineral that's really preferentially depleted in our food supply, and that's copper. And copper is actually required for an enzyme that controls about 270 peptides in the body. And insulin cannot be activated to work in the cell without the PAM enzyme that requires copper. So the herbicide glyphosate preferentially chelates copper all the way down to a pH of 1. It's depleting it from our soil, it's depleting it from our plants, and it's depleting it from our animals. And so I cannot overemphasize the importance of minerals. And one of the things that the keto diet does so well is help us, get away from the foods that are loaded with glyphosate. So a lot of those grains, a lot of things that we don't eat in keto, they now use glyphosate as a drying agent. So at the end of harvest, they'll spray things so that it all dies at an even rate and they can harvest it faster. So things like lentils and chickpeas and wheat and barley, any of those grains, they're using glyphosate. And so when they use it at the end of harvest, it has an even higher load of that. And glyphosate is also an antibiotic. It's been patented as an antibiotic, and it selectively kills the good bacteria in your gut that you need and allows the bad bacteria to exist. I'm sure a lot of people in here are fans of Jason Fung, and he talks about insulin resistance being, it's really more of an overflow issue. So I like this diagram that I got from him, how do we sort of stop that overflow? But the other sort of missing piece of this is that communication between the catcher at the top and the circle at the bottom, which is your cell. That where the arrow meets is where the enzyme action makes the insulin active. So you've got kind of two things going on. You've got an overflow paradigm because you're also trying to produce more insulin because it's not working. Simple carbohydrates, we don't like those in here. But, what are they doing? How do they affect the gut? So pathogenic bacteria love sugar. They love glucose. They love fructose. So the more glucose and fructose you eat, the more you're selecting for the pathogenic bacteria. So you'll get higher blood glucose, you'll get higher endotoxin levels, you'll get a reduction in the diversity of the gut bacteria and what they can do. You get this just overall selection of things that are a problem, gut permeability, and the toxin production. So there's something called glycans, which are basically, it's a sugar molecule. It's a carbohydrate. Well, we know what HbA1c is, or most people are looking at that are keto. So that is basically, that's a molecule that has a sugar added to it. And what they're measuring is how much it is, quote, glycosylated, so it gets those glycans on there. So these simple carbohydrates are what is loading the sugar molecules onto the HbA1c that we're trying to get rid of. But glycans also they do lots of different things, and so they're also sort of feeding the gut lining, so that mucin lining, which is kind of like snot, that's actually fed by glycans. So they're breaking down fibers and making prebiotics and helping to feed that gut lining. So you do need fiber in order to support the lining of the gut, but you want to avoid the simple sugars. Keto diet research is a really interesting area, and after 8 years of being a citizen scientist and digging into the science papers, what I have learned is you really have to read the fine print in these studies, because there seems to be a bit of a concerted effort to try to tell people that keto is not good for you. But if you go into the keto diet research, you will start to see that they're not really feeding the mice and the rats what we consider a keto diet. So I'll show you a little bit about that in a minute. But one of the things that does happen in a keto diet is this increase in BHB, beta hydroxybutyrate, which we're all focused on, and that actually feeds that the thin layer of cells in the gut. So it's very important to protecting that gut lining is to have the beta hydroxybutyrate. And one of the things keto does is also it lowers this inflammatory reaction with the Th17 cells. Those are also upregulated with a strain of bacteria called bifidobacteria. So we do get lower bifidobacteria, but we get this lower inflammatory response. And I know, like in my keto camp group, we've had some discussions about this because you go on keto and your inflammation starts to go away. Well, this is one of the reasons why, because these while they're host defense cells, they're overactivated, and when you go on keto, that overactivation goes away because you're not feeding the bacteria that are feeding that inflammation. So, when you go on very low carbohydrate diet, you get these great benefits that we all know about and talk about. And this is a study actually in endurance athletes where they used the exogenous ketones and really showing what a difference that can make in changing something called muscle glycolysis and plasma lactate concentrations. So these can also affect our brain health. So glycolysis is basically the process of producing energy through fermentation. So again, back to those simple sugars. If you start producing your energy through fermentation, it's not as efficient. So you're not getting as much energy as you would be if you're producing energy from fats. Here's to my myths and facts about ketosis. As I started working on this and trying to pull together some scientific research that was actually feeding the animals in the studies a real ketogenic diet, I could not find one. Most of the diets that I looked at, when you look in the materials and methods, they're actually feeding the mice seed oils or trans fats. And then they're basically saying that a ketogenic diet is not healthy. Well, we know why that is, because that's not really a ketogenic diet. So I say, if anybody brings that up, look at the fine print on the study that they're showing you. So kind of along this path of looking at the microbiome and trying to figure out how to restore the internal pharmacy, my first focus was really in helping my husband. And I went to a conference where they showed that the sugar alcohol mannitol, which is a sugar substitute, could actually stop the aggregation of the proteins that cause Parkinson's. So I came back and started studying this sugar molecule, and we looked to formulate a probiotic that could actually convert glucose and fructose into mannitol and restore this in the gut. Mannitol is actually the most abundant sugar in nature, and we don't use it. Humans don't metabolize it the same way we do other sugars. So when we started to look at it, I just made it for him initially, and he started getting better. And so then we started having other people try it. And along the way, did some studies I'm going to show you. But what we found was that, of course, when you're converting glucose and fructose, you're shifting that sugar profile in the body, and you're changing insulin. And I had, I'm not supposed to say this, but I had somebody with diabetes who got off their insulin. And so we started saying, okay, there's something remarkable to this, and it's stopping that overflow into the cell. So, and it affects glucose metabolism, but it also affects mitochondrial health and overall gut health. And we saw before, if you were in here before, she was talking about circadian clock and the circadian rhythm. Bacteria actually produce melatonin. They have circadian clocks as well, and so it helps support that. So the deeper we started to dive and look at what we had made initially for my husband was that, this has the ability to support ketosis, to enhance energy production, and to kind of change the whole biome. Ken is actually down at our booth. He was someone I knew from my Parkinson's research a long time ago because he worked at the Michael J. Fox Foundation. But he was doing a ketogenic diet and was having trouble. Like he could not fast past noon. He was so hungry, he could not fast past noon. So he did his little N of 1 study with a CGM, and what he found just within a couple of days was that he could fast for longer and longer. And this is his data so he took a month's worth of data beforehand, and then after 12 days on the sugar shift product, his fasting blood glucose had dropped and his ketones had increased. So he was able to get up into a therapeutic level of ketosis. This was one of our early studies we did where we were actually looking at people with IBS type symptoms and before and after, but we were also just using our computational model to look at the genes and to see, okay, did we get the effect that we thought we were going to get? And we got this increase in butyrate production genes, increase in mannitol genes, increase in glycolysis genes, and the siderophores, which affect minerals and mineral uptake in the gut. So, and our kind of surprise, kind of cherry on top, was we got a decrease in antibiotic resistance genes, which is a problem across the globe right now. So I think I mentioned we used computational modeling, so we use machine learning to show how the bacteria interact together and what they do, and this is our output of looking at that. So the overall community increases the production of butyrate, and then we show over to the right-hand side which different species of bacteria are the butyrate producers or the users of that. And then this was a study we did. This is actually my husband's study where we showed there were markers in the before sugar shift that were associated with Parkinson's in our Parkinson's microbiome research. The middle is actually how the product cross-feeds each other, and this is what his gut looked like after, more robust and diverse. And that was 120 days. This phosphoketo is really critical to prebiotic, and we can produce prebiotics in our body. So you can take them, yes, but you can also produce them. And so what this product is actually doing is producing a prebiotic for you in your gut. And you get that, you get the glycan metabolism that's feeding the mucin for the gut lining, you get the butyrate production, and you get glutathione production. So you're getting a detoxification benefit as well. So what are the benefits of the product? Well, it's going to convert that. I mean, we try not to eat glucose and fructose, but there's so many hidden sources of that. And then another thing that I've learned in the last 6 months, Dr. Richard Johnson, who wrote the book Nature Wants Us to Be Fat, he actually talks about in his book when we're under stress, we endogenously produce fructose in our bodies. And so that's something, when I started talking to people who have been doing keto for a long time, and they're like, I don't understand it, I'm not losing weight, I'm not this. And when I read that, it was like this light bulb went off for me, because I'm like, what kind of stress are they under? So it's not just emotional stress, it's back to that nutritional stress. So when we're not getting enough minerals, we're under stress. So we can be producing fructose in our body. And so, we need to get rid of it. You'll get improved energy osmoregulation, so that's that water homeostasis, so improving, bowel regularity. And we've had people on both sides of the spectrum, either chronic diarrhea or chronic constipation, who've had improvement from the improved osmoregulation. I'm going to show you my clinical trial data, but it lowers that serum lipopolysaccharide measurement, so that inflammatory marker, you get the glutathione and the glycan. So we're in the process of publishing a paper. We did a study in a diabetic cohort, and there's they don't do it here in the U.S. as much, but there's an index called the HOMA-IR, which is a measure of insulin resistance or insulin sensitivity. So this, the columns on the right-hand side are actually the changes in HOMA-IR in the people who took the product versus controls. And then those are just some individual case examples from the study. And then this, so our study was 90 days, but at the end of the 90-day study, we unblinded the people and took 10 of the subjects and kept them on the product. Because HbA1c, your blood cells, typical survival is about 120 days. So 90 days is not always a good enough time frame. And a lot of studies where they're looking at HbA1c, 90 days is not enough. So we kept 10 more people on the product for another 3 months, and you can see we had a 14% drop in HbA1c, and the postprandial and fasting blood glucose continued to improve. And this is our measurements of the serum lipopolysaccharide drop, so that inflammatory marker that is dropping over time because we're removing the food source for the gram-negative bacteria. So there's less and less of that inflammatory cell wall. So that's you can take your ketosis to the next level by adding sugar shift and reducing these inflammatory markers and helping to restore your overall gut health. So, do we have any questions? My husband is actually doing extremely well, but I will say he got COVID, a very severe case of COVID at the end of 2021, and had about 6 months of pretty severe long COVID symptoms, and we started doing the Prolon fast mimicking diet with him. He can't really, he has trouble just doing a long fast for himself, but the Prolon fast mimicking diet has actually turned his long COVID symptoms around, and he does that about once every 6 weeks. He'll do a Prolon, if you're familiar with them. They are not histamine. These are not histamine-producing bacteria. So, is it going to be taking? So I have people who take it and kind of reset their gut, and then take a break, or they reset their gut and they're happy. I do have, so I have a doctor in Hawaii who, he actually got a virus from one of his patients in the 1970s and suffered from severe diarrhea for 50 years. And he takes this product, and basically it's the only thing that helped him, and when he ran out of it, his symptoms came back. So I think it really depends on what your underlying ecosystem is, whether you need to take something, long term, or whether you can kind of reset your gut. And a lot of it also depends on kind of what sort of food are you putting in? What are you putting in the pipe? Because at the end of the day, that's feeding your bacteria. What about yogurt? So, that's interesting too, because actually I have a little mini fan club of people from Dr. William Davis, the Wheat Belly doctor. And he has a protocol in his Super Gut book where people treat SIBO, small intestinal bacterial overgrowth, using a specialized yogurt formula that he makes. But they also make a yogurt with my sugar shift formula. So you can actually just take 4 capsules and make a plain yogurt with the sugar shift formula. So I do have quite a few customers who do that. And in fact, I'm actually getting ready to make just a powder product for people to make yogurt. But I recommend if you're going to make a yogurt with a dairy product, that you make sure that it is a grass-fed, organic, high-fat yogurt. Does it help from detoxing sugar and sugar cravings? Customers do tell us that it does break their sugar cravings, but I also have people who have had candida. So if you're trying to get rid of candida, how when you start to remove the sugar, it fights back and makes you want more. So I have had people in that transition period where, they maybe had a week or two where they were like, it's not working, because they'll fight back. That's a good question, and I'm not sure I have the answer. But I do have a lot of people who tell me, like, my cravings are gone in a day or 2 days. So, I think a lot of that depends on what and then the other detox question. So I have a strain of bacteria. It's my Lactobacillus plantarum that's in my formula is actually a formula that we prospected ourselves. It came from an elderberry tree in my neighborhood. And it was, most lactobacillus are not resistant to glyphosate, so it kills lactobacillus. But this strain of bacteria was resistant to glyphosate, and we sequenced its genome, and we found that it had something called the third pathway that can break glyphosate all the way down to carbon, phosphate, and water. So we use that actually in all of our products, and then we also have an agriculture company where we're working to clean up the glyphosate in the soil using our microbes to do that. Well, most people are depleted in copper generally because I think we get in a normal diet like less than a milligram of, I think it's like 0.8 milligrams of copper a day, and you need 2. You don't need a lot, but it's very critical to these enzymes and the recycling of iron in the body. So most people are going to be depleted of it. Of course, if you're eating keto and you're eating beef liver or you're taking a grass-fed beef liver, but there's also been some interesting research on some of the grass-fed beef liver products not having as high a copper content because glyphosate, it's in the air, it's in the water, it's everywhere. So if the farmer next door is spraying glyphosate on his crops, it bleeds down into the water that is watering everything. So, and it's coming down in the rain. There's a Dr. Stephanie Seneff, who's a researcher at MIT, has written a lot about this and shown that it's actually in the air, in the rain, in the water, in the rivers. And so that's actually one of my personal missions, is to clean that up and to do that through our soil probiotics and our human probiotics. So I do actually recommend a supplement for the copper. I take a copper supplement called Recuperate. The other thing that's really important to help copper properly load into the proteins in your body is whole food vitamin C. So you hear people all the time talking about vitamin C, vitamin C. Most vitamin C that is in the supplement market is ascorbic acid. Ascorbic acid actually depletes copper. You need whole food vitamin C that is ascorbate. And that molecule helps the copper to properly load into the proteins in the body and is very important. So we do have people who are measuring ketones who report to us their ketones do go up. And you can talk to Ken, who did that study at our booth, and he can sort of tell you his experience. It is a way to tell if the products work, one way to tell. The insulin resistance, that takes a bit longer. So that, cleaning that whole thing up is really more like a 120-day process. Recuperate does not have zinc in it, but you have to be careful with zinc because you don't want to take a bunch of zinc all the time if you're not taking zinc and copper paired together, because because they use the same receptors in the body, and so then zinc can bind up all the receptors, and then the copper can't get in. Yeah, I mean, yeah, you probably don't want to take like 50 milligrams of copper a day. Yeah, so, yeah. And I think it's always best if you can get your nutrients through whole foods, but there's such a challenge with getting what we need from the food supply right now. Anything else? Okay. Thank you so much.